Module 3, Cell Division and the Cell Cycle. Terminology and basic principles. Copy number. Different cells can vary in the number of copies of each DNA molecule. The term ploidy describes the numbers of copies, N, of the basic chromosome set, and also describes the copy number of each of the different nuclear DNA molecules. The DNA content of a single chromosome set is represented as C. Human cells and the cells of other mammals are mostly diploid, or 2C, with nuclei containing 2 copies of each type of chromosome, one which is paternally inherited and one which is maternally inherited. Sperm and egg cells are haploid cells that contain only one of each kind of chromosome, and thus they are 1C. Human sperm and eggs have 23 different types of chromosomes, and so N equals 23 in humans. Some specialized human cells are nulliploid, or 0C, because they lack a nucleus. Examples of these include erythrocytes, or red blood cells, platelets, and terminally differentiated keratinocytes. Others are naturally polyploid, more than 2C. Polyploidy can occur by 2 mechanisms. The DNA might undergo multiple rounds of replication without cell division, as when the large megakaryocytes in blood are formed. They have 16 to 64 copies of each chromosome, and as a result, the nucleus is multilobed. Alternatively, polyploid cells can originate by cell fusion to give rise to cells with multiple nuclei, as in the case of muscle fiber cells. Polyploidy is also a hallmark of cancer cells. In cancer cells, DNA synthesis is dysregulated, and as a result, the nuclear DNA content increases, giving rise to large multilobed nuclei. Mitochondrial DNA copy number. Whereas all diploid cells contain 2 copies of each nuclear DNA molecule, the number of mitochondrial DNA molecules can vary from hundreds to many thousands according to cell type, and in some cases can vary over time. The 2 types of haploid cells that show very large differences in their mitochondrial DNA copy number are the human sperm and egg. A human sperm typically has about 100 mitochondrial DNA copies, but a human egg cell usually has about 250,000 mitochondrial DNA copies. Gametes. The germline is the collective term for all cells that pass genetic material from one generation to the next. It includes haploid sperm and egg cells, which are known as gametes, and all the diploid precursor cells from which they arise by cell division, going all the way back to the zygote. The non-germline cells of the body are known as somatic cells. So we have 2 types of cells, the gametes and the somatic cells. In humans, where N is equal to 23, each gamete contains 1 sex chromosome plus 22 non-sex chromosomes, which are referred to as autosomes. So we have the sex chromosomes and the autosomes, which are non-sex chromosomes. In eggs, the sex chromosome is always an X, whereas in the sperm, it may be an X or a Y. Changes in chromosome and DNA content during the cell cycle. During normal periods of growth, there is a need to expand cell number in the fully grown adult. The majority of cells, however, are terminally differentiated and do not divide. But stem cells and progenitor cells continue to divide to replace cells that have a high turnover, notably those in the blood, skin, sperm, and intestinal epithelial cells. Each round of the cell cycle involves a phase in which DNA replication, the S phase for synthesis of DNA, and a phase where the cell divides, the M phase. Note that the M phase involves both nuclear division, mitosis, and cell division, or cytokinesis. In the intervals between these two phases, there are two gap phases, the G1 gap phase, the gap phase between M phase and S phase, and G2, the gap phase between S phase and M phase. This is shown in the figure by the circular diagram at the lower right-hand corner of this slide. In addition to the G1, S, G2, and M, we also have a G0 phase where cells can stop cycling for a period of time. It is a quiescent phase, and when conditions become suitable, cells may move from this G0 phase to re-enter the cell cycle. In the expanded panels on this slide for M and S phases, we show, for convenience, just a single chromosome, and we illustrate in the S phase panel, which is on the left-hand side of this slide, how a single chromosome relates to its DNA molecule at different stages. Chromosomes contain one DNA double helix from the end of M right through and just before they duplicate in S phase. After duplication, the two double helices are held tightly together along their lengths by binding proteins known as cohesions. And the chromosome now consists of two sister chromatids, each having a double helix. The sister chromatids become more obvious in late S phase, when most of the cohesions are removed except for some at the top of the centromere, which continue to hold the two sister chromatids together. And then, as shown in the upper right-hand corner of the slide, during M phase, the sister chromatids finally separate to form two independent chromosomes that are segregated into daughter cells. Note that the S phase chromosomes in the left panel are shown purely for convenience, in a compact form, but in reality they are enormously expanded. This slide depicts the unequal replication of individual mitochondrial DNA. Unlike the nucleus, where replication of chromosomal DNA molecules normally produce two copies, mitochondrial DNA replication is stochastic. When mitochondria increases in mass in preparation for cell division, the overall amount of mitochondrial DNA increases in proportion, but individual mitochondrial DNAs replicate unequally. In this example, the mitochondrial DNA with the green tag fails to replicate, and the one with the red tag replicates to give 3 copies. Variants of mitochondrial DNA can arise through mutation, so that a person can inherit a mixed population of mitochondrial DNAs, which is known as heteroplasmy. Unequal replication of pathogenic and non-pathogenic mitochondrial DNA variants have important consequences in human health, as will be discussed later in this course. This slide shows mitosis, or nuclear division, and cytokinesis, which is cell division. Several stages are part of this process, and they include and begin with prophase. And early in prophase, the centrioles, or short cylindrical structures composed of microtubules and its associated proteins, begin to separate and migrate to opposite poles of the cell to form the spindle poles. In prometaphase, the nuclear envelope breaks down, and now the highly condensed chromosomes become attached at their centromeres to the array of microtubules extending from the mitotic spindle. At metaphase, the chromosomes all lie along the middle of the mitotic spindle, still with the sister chromatids bound together because of residual cohesions at the centromere that hold the duplicated DNA helices together. Removal of the residual cohesions allows the onset of anaphase. The sister chromatids separate and begin to migrate towards opposite poles of the cell. The nuclear envelope forms again around the daughter nuclei during telophase, and the chromosomes decondense, completing mitosis. Before the final stages of mitosis, and most obviously at telophase, cytokinesis begins with constriction of the cell that will increase progressively to produce two daughter cells. Meiosis. Meiosis is a reductive division undergone by premadural spermatocytes and oocytes. The process differs in sperm versus oocyte. The primary oocyte will give rise to one mature oocyte and a polar body, whereas the primary spermatocyte will give rise to 4 spermatozoa. Prophase stages in meiosis. There are several different stages of meiosis that occur in prophase that give rise to genetic diversity of the gene pool. As shown in A, in Lipotine, the duplicated homologous chromosomes begin to condense but remain unpaired. In this particular diagram, the maternal homologs are colored pinkish red, whereas the paternal are shown as blue. Production of gametes. One of the features of the production of gametes is the generation of genetic diversity. One of the ways that this is achieved is through the process known as recombination. The prophase of meiosis I begins during fetal life, and human females can last for decades. During this extended process, parental and maternal chromatids within each bivalent normally exchange segments of DNA at randomly positioned but matching locations. This process, called recombination or crossover, involves physical breakage of the DNA in one paternal and one maternal chromatid and the subsequent joining of maternal and paternal fragments. The sole purpose of sex in biology is to produce novel combinations of gene variants, and the instrument for achieving this aim is meiosis. The whole point of meiosis is to produce unique gametes by selecting different combinations of DNA sequences on maternal and paternal homologs. Although a single ejaculate may contain hundreds of millions of sperm, meiosis ensures that no two sperm will be genetically identical. Equally, no two eggs are genetically identical. Each zygote must also be unique because at fertilization, a unique sperm combines with a unique egg. However, a unique fertilization event can occasionally give rise to two genetically identical or monozygotic twins if the embryo divides in two at a very early stage in development. Monozygotic twins are nevertheless unique individuals. Genetics isn't everything in life. Independent assortment of maternal and paternal homologs. This figure shows a random selection of just 5 of the 8,388,608 theoretically possible combinations of homologs that might occur in haploid human spermatozoa after meiosis in a diploid primary spermatocyte. Maternally derived homologs are represented by pink boxes and paternally derived homologs by blue boxes. For simplicity, the diagram ignores recombination. Recombination superimposes additional genetic variation. This figure illustrates the contribution to genetic variation at meiosis I made by independent assortment of homologs, but for simplicity ignores the contribution made by recombination. In reality, each transmitted chromosome is a mosaic of paternal and maternal DNA sequences, as shown here.